Hi everyone, welcome to another video of the Science Club. In today's video, we are going to quickly learn about Hershey and Chase experiment. This experiment helped to confirm that the genetic material of the cell is the DNA. In the early 19th century, the scientific community was trying to identify the hereditary material inside cells. Griffith's experiment in 1928 was a stepping stone for the discovery of the genetic material. Several years later, Avery and his team isolated DNA from cells and showed that the genetic material inside the cells is nothing but the DNA. But these results were not accepted by all until Hershey and Chase published their experimental results in 1952. The scientific community later generally acknowledged that DNA was the genetic code material. This discovery led to a more detailed investigation of DNA to determine its composition as well as its 3D structure. Using X-ray crystallography, the structure of DNA was discovered by James Watson and Francis Crick shortly thereafter in 1953. Griffith, while searching for a cure for pneumonia, discovered bacterial transformation instead. He showed that non-virulent bacteria could be transformed into virulent bacteria by means of what he called the transforming principle. This transforming principle carried the genetic information. However, what exactly was this transforming principle? Was it the DNA or the protein or something else? Maybe polysaccharides. Avery, McLeod, and McCarthy showed that it was the DNA that led to the transformation of one strain of bacteria to another as observed by Griffith. The results of these experiments provided evidence that DNA was the biomolecule that carried genetic information. The results were met with skepticism and many still believed proteins to be the hereditary material. DNA with 4 nucleotide bases was considered too simple to be a molecule carrying the genetic information. Later, Hershey and Chase took efforts to find out the biochemical nature of the genetic material inside cells. For these experiments, the key element was a simple organism called the bacteriophage. What exactly is a bacteriophage? Bacteriophages are nothing but viruses that infect and replicate inside bacteria. The virus doesn't have their own mechanism of reproduction. So, in order to multiply, they depend on a host organism. Here, in case of bacteriophages, bacteria are their host. How exactly does a bacteriophage infect bacteria? The bacteriophage attaches to the host bacteria from the outside. Once they attach to the host cell, their viral genetic material is transferred to the host bacteria. The infected bacteria now start to replicate the viral genetic material. In the process, new bacteriophages are made inside the bacteria, eventually killing the bacteria. Hershey and Chase conducted an experiment to discover whether it was protein or DNA that acted as the genetic material that entered the bacteria. It was clear that the viral material inserted inside the bacteria contains the information for creating new viruses and is therefore the hereditary material. But what exactly is the nature of this material? Is it made of nucleic acids or proteins? To answer this question, Hershey and Chase devised a simple experiment. Viruses were known to be composed of a protein shell and DNA. So they chose to uniquely label each with a different radioactive isotope. In the first group, radioactive sulfur was used to coat the protein sections of the bacteriophage because sulfur is contained in protein but not in DNA. In the second group, since phosphorus is contained in DNA but not in proteins, radioactive phosphorus 32 was used to label the DNA contained in the phage. The labeled phages were then allowed to infect bacteria. After the infection had occurred, they wanted to check whether the radioactivity was found inside the bacteria or outside. For this, firstly, they used a blender to remove the bacteriophages that were already attached to the outside of bacteria. Once separated, this mixture was then centrifuged. Since bacteria are heavier than the small viruses, they collected at the bottom of the tube and made a cell pellet. The results of the experiment were very clear. In the case of group 1, when the bacteriophages with radioactive protein were allowed to infect bacteria, most of the radioactive signal remained outside the bacterial cells. So the proteins are not the ones getting inherited. Whereas in case of group 2, where bacteria were infected with bacteriophages containing radioactive DNA, most of the radioactivity after the infection was found inside bacterial cells. This proves that it is the DNA that is getting transferred inside the bacteria and is therefore the genetic material that the virus is injecting. This simple experiment provided Hershey and Chase with the pivotal evidence to claim that it is the DNA and not the protein that is the genetic material that is being inserted and passed on to the next generation.